And we would like to thank Sir joining us all the way from UK. And the stage is yours. The total time allotted is 25 minutes. Thank you so much.
Good morning, everybody.
Could you just take the mic down towards yourself? Yeah, perfect. Thank you. Better? Yes.
Good morning. Can you hear me okay? Right, absolute pleasure to be here, and I'd like to thank the organisers for inviting me to come and speak to you today. So my job is to talk to you about the kind of basics of immunology that is of relevance to transplantation. And I've also been asked to talk about how you deal with a multi-ethnic transplantation population. So I'm going to do that in those in that order. So I am normally based in Leicester. I'm doing a sabbatical at the moment in Bermuda, which is the lovely hospital we have there. And what I'm going to talk about today really is what I've been doing in Leicester for most of my consultant career. So we're going to talk about the very basics to start with. So you all know that transplantation is the process of taking cells, tissues or organs from a donor and placing them in a different individual. And that can either be in the normal position, which is an orthotopic transplantation, or in an anatomically different position, so a heterotopic transplantation, which is what we would normally do with a kidney transplant, for instance. And then in terms of the relationship between the donor and the recipient, there are a number of terms that we use. So autologous means you transplant tissue within the individual. So the only time we'd really probably do that is an autotransplantation of a kidney in somebody with resistant renovascular hypertension, for instance. Syngeneic is a transplant between genetically identical individuals. Allogeneic, which is what we're going to be talking about mostly today, is a graft between two genetically different individuals. And xenogenetic is transplantation across species barriers. So it's nearly 70 years since the very first organ transplant. So it was done in 1954 in Boston, and that was a syngeneic transplant, only possible in those days because it predated the advent of immunosuppression. And it was, as I say, between two identical twins. The recipient survived 8 years and died of unrelated causes, and the donor survived for 56 years afterwards. So the general principle of transplantation is that transplantation of a graft between two genetically different individuals will lead to graft failure through a process called rejection, which is related to an adaptive immune response. And the graft is identified as foreign because of the differential expression of proteins on the cell surface. And in the normal transplant situation, we refer to those as alloantigens, or if it's cross-species, to xenoantigens. Now, the immune response that we see in this situation is exactly the same as you tend to see in the response to a microbial infection. And it's predominantly an adaptive immune response, which involves both the cellular and the humoral arms of that response. And there's also a contribution from the innate immune system as well. So what are the alloantigens in human transplantation? So the predominant ones are antigens produced by the major histocompatibility complex, or in humans, known as the human leukocyte antigen family, or HLA. And the main purpose of these antigens is to bind to present peptides to T cells. There are a number of minor histocompatibility antigens, which I'm not going to dwell on because I know there's a talk specifically about this later today, but that just gives you some of the examples of minor antigens that have been identified as having implications for transplant survival. So I'm going to talk a bit more detail about HLA, and I know, again, there's going to be more detailed talks about this during the course of today. So the MHC region is a zone of highly polymorphic genes, which is found on the short arm of chromosome 6. And its main function is to present antigens to antigen-specific T cells. And it's important to say that T cells will only respond to foreign antigen when it is presented as peptide fragments, which is complexed with MHC. So that relationship is critical to the immune response. And for the purposes of transplantation, there are two main groups that are of relevance. So there's class 1 and there's class 2. There's also a minor group, class 3, which has relatively minor implications in terms of transplant survival. So again, I know that there's going to be a more detailed talk about HLA in a bit, but this just shows the structure of the HLA genes on chromosome 6. So this is the start of the chromosome here, base pair 0. So you've got class 2 region to start with, class 3 region in the middle, and then the class 1 region going up to about 4,000 base pairs up the chromosome. Again, this is going to be covered. I'm just going to mention this very, very briefly. So this just shows some of the complexity that has been demonstrated in the HLA genes over the years. So historically, HLA was identified using serological techniques, so antibodies that recognize the different structures of the various HLA. So these are the serological types here, and you can see there's relatively few of them. With more detailed sequencing processes, it's been identified that within each HLA category, although there are very few serological types, there are many, many more protein types. And those are encoded by almost twice as many different alleles. So the complexity of the HLA genes is immense. And in terms of transplantation, the critical thing is what the immune system sees as foreign. And again, the nomenclature of HLA genes will be covered a bit later, but this just shows the kind of modern context of it. So you've got the HLA prefix, then you've got A, which is the gene, then you've got O2, which is the allele group, so that will be HLA2, and then you've got another determinant of what the specific HLA protein is. And then right at the end, you've got a couple of modifiers that tell you about genetic mutations that arise in the exonic and intronic regions. And then at the very end, there's a suffix that explains how those mutations affect the expression of those HLA proteins. So I'm just going to talk very briefly about the structure. So this is class 1 HLA, so that's HLA B, A, B, and C. So it's a heterodimer of an HLA protein, an alpha chain, which is linked to beta-2 microglobulin non-covalently. And the critical part of the MHC molecule is at the top here, the area between the alpha-1 and the alpha-2 loops, which is the peptide binding cleft. This is just a ribbon diagram. This is HLA-B27, in fact, and you can show the peptide binding groove in between the alpha-1 and alpha-2 chains. So the critical thing about class 1 HLA is it presents cytoplasm-derived peptides, either normal components of the intracellular environment, but more usually parasites, typically viruses. The proteins are degraded within the cells, and then they are eventually assembled with the MHC complex and expressed on the cell surface. And class 1 MHC specifically presents antigen to CD8 positive T cells. And those activated CD8 cells induce apoptosis or cell death through the release of cytotoxic proteins. So HLA class 1 is expressed on all cell types except red blood cells. And there's also high expression levels on professional antigen-presenting cells, typically dendritic cells and macrophages. So class 2 HLA, so that's HLA-DP, DQ, DR, slightly different conformation. So the genes for class 2 encode two different peptide chains, the alpha and the beta chain, and the peptide binding cleft is between the alpha-1 and the beta-1 loops. And again, this just shows you the ribbon diagram for, I think this is HLA-DR1. And again, you have the peptide groove between those two chains, which is the critical part for antigen presentation. So in contrast to class 1, class 2 binds peptides derived from extracellular proteins, and the alpha and beta chains are associated in the endoplasmic reticulum and then expressed on the cell surface, where it interacts specifically with CD4 positive T cells. Now, class 2 is more confined in terms of its expression. It is constitutively expressed on dendritic cells, macrophages, and B cells, but on epithelial and endothelial cells, its expression is upregulated by cytokines, particularly IL-2 and gamma interferon. So how are these foreign peptides recognized? So what are the mechanisms of allorecognition? So there are two well-defined pathways. Hasn't come out quite right. So the direct pathway is where the T cell recognizes donor MHC that's bound peptide on the dendritic cell from the grafted cell. So this is donor HMC expressed on a donor. Sorry, this has all got mixed up. I'm sorry about that. Anyway, so the thing about direct pathway is that it is donor antigen-presenting cells, typically dendritic cells, which are passenger cells when the transplant is donated, and they express, they demonstrate intact MHC in association with a peptide, and it doesn't matter what the peptide is, but it expresses or shows the MHC to CD4 positive and CD8 positive T cells, and then you get a downstream response from that. So for the direct pathway, non-peptide bound MHC is unstable, and therefore the peptide is co-expressed, and it doesn't really matter what that peptide is. The pathway is unique to allorecognition. It's very powerful and is the cause of the very, very early high post-transplant rejection risk, and that's caused by the passenger antigen-presenting cells, typically dendritic cells. The other thing that's important is there's a very, very high frequency of T cells that inherently recognize any single MHC component. And it's thought that 1 to 10% of T cells will be able to mount response to foreign MHC. And antigen-presenting cells are also able to provide the necessary co-stimulatory signals as well. The risk of rejection related to this falls as the passenger antigen-presenting cells become depleted. The second pathway is the indirect pathway. So in this situation, it is mediated by recipient antigen-presenting cells. So you have allogenic MHC expressed on the cell surface, which is endocytosed. The peptide fragments are expressed in the context of class 2 MHC, 2 CD4 positive T cells, and the downstream response of that is the generation of CD4 positive T follicular helper cells, which is necessary for antibody production, or CD4 positive helper T cells, which is related to cytokine-mediated graft injury. And the key thing there is that this is the normal mechanism of antigen recognition. So you've got allo-MHC recognized by self-antigen-presenting cells. So the dominant imagenetic proteins are the hypervariable regions of donor MHC. The allopeptide reactive T cells are present during both acute and chronic rejection. The other thing to be aware of is the initial immune response is directed against a limited repertoire of MHC allopeptides, but the later responses are related to a broader repertoire of allopeptides, which is a process known as epitope switching or spreading. So T cell activation requires 3 distinct signals. I've talked to you about signal 1, which is the presentation of foreign antigen in the context of MHC. The second one is the interaction between cell surface receptors with specific ligands on the antigen-presenting cells. So broadly speaking, there are 2 main groups, the CD28 and B7 signaling. So normally CD28 will interact with B7-1 or B7-2, and that will cause activation of naive T cells. It can also interact with CTLA-4, with B7-1, B7-2, which kind of provides a break to T cell activation. And then there's a couple of other members of the B7-CD820 family, which again provide competing impacts on the activation of T cells. This has been targeted therapeutically, so abatacept and belatacept have been developed as immunosuppressants, so we are aware of belatacept being currently licensed for use, and it basically is a CTLA-Ig fusion peptide which helps to inhibit T cell activation. The second signal is through CD40 ligand. So T cells initially recognize antigen with or without B7, which causes expression of the CD40 ligand, which interacts with CD40. That causes activation, which causes release of cytokines from the dendritic cells, and then that signal causes proliferation and differentiation of the T cells. And then the third signal is in 2 parts. The first is T cell proliferation related to interleukin-2, and there are differential effects of interleukin-2 depending on the point in which it occurs. So interaction with a naive T cell, interleukin-2 causes proliferation of effector T cells. Dendritic cell activation of an activated T cell, in contrast, interleukin-2 causes production and proliferation of regulatory C cells. So it provides a break to T cell activation. And the other part of the third signal is the secretion of different cytokines which allow the T cells to differentiate into different groups. So just as a brief example, IL-12 tends to drive the production of Th1 cells, IL-4 tends to drive the production of Th2 cells, but it's a way more complex phenomenon. This just shows the kinetics of T cell activation. So you've got a naive T cell. When it gets activated, you get cell signaling intracellularly. That causes the production of IL-2, the expression of CD69, the IL-2 receptor alpha chain is expressed, and then CD40 ligand is expressed. So in this phase, you get proliferation of the T cells, and then through the production of other cell surface modulators, particularly CTLA-4 and PD-1, you get downregulation of the T cell proliferative response, and cell division starts to die away. Finally, there's a series of signal transduction mechanisms within the cell, which are what are used as the targets of immunosuppression. So just as example, interaction of the T cell receptor with MHC causes release of intracellular calcium, which stimulates calcineurin, and the release of various transcription factors, and that's the target of both calcineurin inhibitors, so cyclosporin and tacrolimus. Sirolimus interacts with the downstream events of interleukin-2 engagement, interleukin-2 receptor engagement, and IL-2 receptor antagonists such as basiliximab obviously causes disruption to the interaction between IL-2 and the IL-2 receptor. In the interest of time, I'm just going to skip through this, and I'm just going to now talk about the effects of multi-ethnicity of transplantation, because this is kind of important. So I just want to kind of talk about this in the remaining 10 minutes. So there are a number of issues around multi-ethnicity in transplantation. So the first thing to say is that the expression of HLA varies according to the racial background. So this is a very complex slide, and I'm not going to go through this in detail, but this basically shows it's a heat map showing the expression of various HLA alleles. This is class 1, HLA A and B. So those are expressed across the bottom here, and it just shows the expression in different geographical populations, which are kind of highlighted down the side here. And it just shows that across different populations, the expression of various alleles varies enormously. So if you then have a migrant population that comes into a population where there's a different expression pattern of HLA, it can cause issues. And you see the same pattern with HLA-DRB. And a very nice place to kind of see this in a bit more usable detail is a website called allelefrequencies.net. So this just is showing the frequency of expression of the allele for A24. So if you look at India down here, there's a reasonably high expression level of HLA-A24. If you look at the United Kingdom, there tends to be much lower levels of expression. So migrant population from West India, which we tend to have in the Leicester area, will tend to express a different cohort of HLA, which will have issues in terms of matching. We know that HLA matching is an important determinant of transplant outcomes. So just one example of this. So this is data from the collaborative transplant study between 2005 and 2014, in the days of modern immunosuppression, and it still shows that for deceased donors, there is an impact of HLA matching in terms of longer-term graft survival. So the difference between 0 mismatch and 6 antigen mismatch is about 10% survival at 5 years. And this just shows how that can impact according to the racial background for a patient. So this is a study from the United Kingdom. Again, busy slide. I just want to highlight one thing. So this demonstrates the HLA mismatch level across different ethnic backgrounds. So level 1 is a very low level of mismatching. So what you can see is that the level of mismatch is way lower in the white recipient population compared to either the black or the South Asian population. So that's in the UK population. So the other issues that will impact on this is there's a higher prevalence of chronic kidney disease in ethnic minority populations, and that there's generally a lower rate of deceased donation in ethnic minority populations. So again, this is UK data. So this just shows the prevalence of end-stage kidney disease in the United Kingdom. So in the white population, so 74% of the end-stage population is made up of white people, about 14% Asians, and about 8.5% black. The UK 2021 census suggests that the black population comprises 4% of the population and the Asian about 9.3%. So there is a significant overrepresentation of ethnic minorities in the end-stage population. And if you look at the transplant wait list, it's even more significant. So the black population represents 12% and the Asian population 18%. And we see a similar pattern in the United States, and I won't go that in any detail, but basically for the black population in the United States, they're overrepresented in the end-stage population, they're overrepresented on the transplant wait list, they tend to wait longer for their transplants, and they tend to do badly while they're waiting for their transplants. As I mentioned, people from ethnic backgrounds, for a variety of reasons, are less likely to be deceased kidney donors. So again, this is a UK study looking at the proportion of deceased donors. So the vast majority were from white donors, 97%. Oops. Only 1.1% were from the black population and 1.7% from the South Asian population. And again, I just remind you of the figures for the UK census and the transplant wait list. So there's a huge disparity between donors from ethnic minority backgrounds and the people who need those kidneys. There is also an impact of organ allocation policies. So this is the old 2006 kidney allocation scheme, and you can see that the priority in terms of allocation is for 000 mismatches or HLA-DR homozygous. And then when you get through those, you get down to the other patient populations. So it was given that you're much less likely to get a well-matched kidney if you're from a minority ethnic population, this disadvantages you. So in 2019, the United Kingdom changed the allocation scheme, and there are now just two tiers. So the first tier is patients with a matchability score of 10. So these are people who are very, very difficult to match, and this is based on their HLA expression and their presensitization levels. It also priority patients with 100% calculated reaction frequency, or it also favors people who have spent a long time, more than 7 years, on the transplant wait list. The second tier is all other eligible patients. And there is now a complex algorithm for working out who gets the priority, and it's based on these combination of factors. So the waiting time is important, donor-recipient risk index combinations, HLA match and age combined, location of patient relative to donor, matchability, donor-recipient age mismatch, total HLA mismatch, and the blood group match. The other thing that's been done is that they've defaulted rare HLA specificities to the more common equivalent. So in basic broad terms, if you have a rare HLA type, so for instance, HLA-A36, which is rare, has a common equivalent of A1. So essentially, if so in terms of some kind of serological issues, A36 is unlikely to be recognized as foreign in a recipient that is A1 positive. So again, that just allows people with rare HLA types to be able to receive kidneys from a non-ethnic matched population. So it's relatively early to tell, but this just shows some of the very early data in terms of the impact on this in terms of ethnic allocation of kidneys. So between April 18 and March 2019, so this was the constitution of the kidney recipients. So 73% were white, 15% were Asian, and 8% were black. And after the introduction of the allocation scheme, the biggest changes in the black population where the percentage of patients has risen from 8% to 10%. And there's a kind of concurrent reduction in the white population. In the United States, they did a similar thing in 2014. So they made changes designed to balance equity and utility, and that just lists some of the things that they did. And again, this did seem to have an impact in terms of kidney allocation to minority ethnic groups. So in this, I just want to highlight the area within the red here, which just shows the ethnic breakdown of who got kidneys before and after the introduction of that allocation scheme. So in terms of African Americans, before the allocation of the screen, 31.5% of patients made up the recipients, and after the introduction, that increased to 36.8%. So it does seem to have some impact on the allocation to ethnic minority groups. So in conclusion, ethnic minority populations are at an immunological disadvantage with respect to transplantation. The differences in HLA expression, higher prevalence of end-stage kidney disease, and an apparent reluctance to donate in minority ethnic populations underlie this disadvantage. Changes to kidney allocation policies can upset some of this disadvantage, and so overall it requires a broader approach to mitigate the increased risk of end-stage kidney disease in minority ethnic populations, and also to look at the societal factors that dissuade people from minority ethnic groups from donating. So sorry that was a little bit rushed, but thank you very much for your attention.